Amino acids contain nitrogen that cannot be stored. Breakdown of amino acids generates ammonia. Some ammonia is excreted in urine. Most converted to urea through urea cycle in the liver. Carbon skeletons used as intermediates in various pathways. Today, we will cover 4 topics. First, we will look at digestion of dietary proteins. Proteins are too large to be absorbed. They must be hydrolyzed first. Hydrolysis is accomplished by enzymes from 3 different organs. Stomach secretes pepsin. Pancreas secretes zymogens that are converted to trypsin, chymotrypsin, elastase, and carboxypeptidases. Small intestine produces enteropeptidase, aminopeptidase, and dipeptidases. Trypsin, chymotrypsin, elastase, pepsin, and enteropeptidase are endopeptidases. They break peptide bonds of non-terminal amino acids. Aminopeptidase and carboxypeptidases are exopeptidases. They break terminal peptide bonds. Digestion of proteins begins in the stomach. As we know from GI physiology, stomach secretes gastric juice that contains hydrochloric acid and proenzyme pepsinogen. Hydrochloric acid serves to kill some bacteria and denature proteins. Pepsin is the enzyme that cleaves proteins, especially collagens, into peptides and a few amino acids. Pepsin is an endopeptidase. It is secreted as pepsinogen, activated by acid or pepsin. As we have discussed in GI physiology, the GI hormone CCK stimulates release of zymogens from the pancreas. Secretin stimulates the pancreas to release bicarbonate to neutralize pH of the intestinal contents. This represents a small intestine, and these are zymogens. Zymogens secreted from pancreas are activated by enteropeptidase in the small intestine. First, enteropeptidase converts trypsinogen to trypsin. Trypsin then converts trypsinogen and all other zymogens into their active forms. Here is a clinical correlation: pancreatic failure. In this condition, digestion is abnormal when pancreas fails to secrete normal amounts of enzymes. It could be caused by pancreatitis or surgical removal of pancreatic head. Without pancreatic enzymes, 60% of fats and 30 to 40% of proteins and carbohydrates are not absorbed. Presence of excess fats in stool is called steatorrhea. Oligopeptides continue to be digested by exopeptidases. These enzymes catalyze the cleavage of the terminal or the penultimate peptide bonds and release amino acids and dipeptides. There are 2 groups of exopeptidases. One is aminopeptidases. They cleave peptides from the amino terminus. They are produced by the small intestine and reside on the luminous surface of the small intestine. Many of these enzymes are zinc metalloenzymes. Second group is carboxypeptidases A and B. They are produced by the pancreas and activated in the small intestine. Now, let's move to part 2. We will look at absorption of amino acids and dipeptides. Free amino acids, dipeptides, and tripeptides are absorbed by intestinal epithelial cells. Dipeptides and tripeptides are cleaved in the cytoplasm of enterocytes by dipeptidases and tripeptidases respectively before entering the portal system. Only free amino acids are found in the portal vein. Absorption of amino acids and dipeptides is achieved by three transporter systems. Here is intestinal lumen, and here is enterocyte. On the brush border membrane, sodium-dependent symporters uptake free amino acids. On the brush border membrane, a similar proton-dependent symporter updates dipeptide and tripeptides. On the basolateral membrane, sodium-independent transporters facilitate movement of free amino acids to the portal vascular system. Among them, sodium-dependent symporters have clinical significance. There are at least 7 symporters. Let's focus on these two. One is neutral amino acid symporter that transports amino acids with aromatic and hydrophobic side chains. They include phenylalanine, tyrosine, tryptophan, methionine, valine, leucine, and isoleucine. Basic amino acid symporter transports lysine, arginine, and cysteine. Here is a clinical correlation. First condition is Hartnup disease. It is an inherited disorder with intestinal defects of amino acid absorption and urinary wastage of nonpolar and neutral amino acid, especially tryptophan. Symptoms are very similar to pellagra, characterized by diarrhea, dementia, and dermatitis. Second condition is cystinuria. It is the most common inherited disease of amino acid transport. It results from defects in kidney transport system for reabsorption of cysteine, ornithine, arginine, and lysine, summarized in a mnemonic COAL. 4 amino acids are present in the urine, especially cysteine. Cysteine is oxidized cysteine. It precipitates to form kidney stones. Now, let's move to part 3. We will look at removal of nitrogen from amino acids. The alpha amino group of amino acids can be removed by either transamination or oxidative deamination. Transamination involves transfer of the amino group from one amino acid to alpha-ketoglutarate. Products are an alpha-keto acid and glutamate. This reaction is catalyzed by an aminotransferase. All amino acids except lysine and threonine participate in transamination. All aminotransferases require the coenzyme pyridoxal phosphate. This is pyridoxal phosphate. It is a derivative of vitamin B6. An amino group from glutamate is transferred to pyridoxal phosphate. The result is alpha-ketoglutarate and pyridoxamine phosphate. The amino group is then transferred from pyridoxamine phosphate to oxaloacetate. The result is aspartate. Vice versa, this reaction can proceed in the opposite direction. Amino group is transferred from aspartate to pyridoxal phosphate, then to alpha-ketoglutarate. The result is glutamate. This enzyme is named after the amino acid that acts as the amino group donor. In this case, it is aspartate aminotransferase. Alanine aminotransferase, or ALT, catalyzes transfer of amino group from alanine to alpha-ketoglutarate. The products are pyruvate and glutamate. That's why this enzyme is also called glutamate pyruvate transaminase. Glutamate collects nitrogen from alanine. This enzyme is produced by the liver in the highest concentration. Please note, this reaction is reversible. Aspartate aminotransferase, or AST, transfers the amino group from glutamate to oxaloacetate. The products are alpha-ketoglutarate and aspartate. As I described to you earlier, this reaction can also occur in the reverse direction. Nevertheless, the forward reaction has special value in the liver. One of the products from the forward reaction, aspartate, is used as a second nitrogen source for urea cycle. This enzyme is found in a variety of tissues. ALT and AST have been used as diagnostic markers for liver disease. For example, in acute liver damage, such as viral hepatitis, ALT to AST ratio increases. Oxidative deamination refers to removal of amino group from glutamate and formation of alpha-ketoglutarate and ammonium. It is catalyzed by glutamate dehydrogenase. It is a mitochondrial enzyme found in a variety of tissues. Through transamination, followed by oxidative deamination, the amino group of an amino acid is finally released as ammonium, which will be converted to urea in the liver. This reaction is reversible. The reverse reaction is used for synthesis of amino acids in many other tissues. Glutamate dehydrogenase has 4 features. First, it uses either NAD or NADP as a coenzyme. The direction of reaction depends on concentration of products and substrate. Third, it is an allostatic enzyme. Allostatic regulators include ADP and ATP. Typically, ADP is a positive effector and ATP is a negative effector. This enzyme represents a key link between catabolic and anabolic pathways. Now, let's move to part 4. We will look at metabolism of ammonia. Ammonia is extremely toxic to the central nervous system. It can cause coma and death. The body must have a mechanism to remove ammonium from periphery and transport to liver without increasing plasma ammonium levels. In the liver, ammonium is converted to urea. Urea is the main nitrogen-containing substance in the urine. Blood urea nitrogen, or BUN, is a useful marker for diagnosis. Its normal range is from 8 to 20 mg per deciliter. Increased BUN suggests kidney disease, heart attack, and dehydration. Decreased BUN, on the other hand, suggests liver disease, malnutrition, overhydration, anabolic steroid usage, or pregnancy. Ammonium is derived from many sources. The main source is amino acids. Ammonium is generated through oxidative deamination in many tissues, especially liver. Second and third, ammonium is derived from glutamine through renal and intestinal glutaminases. Fourth, in the intestine, bacteria urease breaks down urea to form ammonia. Fifth, from amines in the diet or breakdown of neurotransmitter through monoamine oxidase. Finally, ammonium is derived from degradation of purine and pyrimidine by deaminases. Two systems are used to transport ammonium to the liver. First is glutamine system. Most tissues transport ammonium to the liver by glutamine. Ammonium is converted to glutamine by glutamine synthetase. Glutamine can be easily transported out of cells and taken up by the liver, where ammonium is released by glutaminase. Second is alanine system. In particular, muscles use alanine to convert ammonium to alanine by ALT. Alanine is then transported into liver through transamination followed by oxidative deamination. Ammonium is released in the liver. Here are two key enzymes in the glutamine system. Glutamine synthetase is found in a variety of tissues. Glutaminase is mainly found in the liver and the kidney. Ammonia is converted to urea mainly in the liver. Both urea and glutamine are transported to the kidney, while ammonia is released from glutamine by glutaminase. Both urea and free ammonia are excreted to the urine. Close to 90% of nitrogen in urine is urea. This is urea. One nitrogen is derived from ammonia. Second nitrogen is derived from aspartate. Carbon dioxide provides carbon and oxygen. Urea is synthesized mainly in the liver through urea cycle. This is the position of the urea cycle on the metabolic map. Urea is a small compound. As we have seen in breakdown of acetyl-CoA in TCA cycle, when we deal with a small compound, we do not have a simple way to make it or break it. Instead, we borrow a compound, put all elements on it, and then we break it. The compound we borrow in urea cycle is ornithine. This is ornithine. The carbonyl group and first nitrogen are derived from bicarbonate in ammonia. They are converted to carbamoyl phosphate by CPS1. It stands for carbamoyl phosphate synthetase 1. Energy is derived from 2-ATP. Then ornithine is joined with carbamoyl group by OTC. OTC stands for ornithine transcarbamylase. The result is citrulline. These two reactions take place inside the mitochondria. Citrulline is transported into the cytoplasm where it is joined with aspartate by ASS. ASS is argininosuccinate synthetase. This reaction consumes a third ATP. Argininosuccinate is cleaved to release arginine with fumarate as byproduct. It is catalyzed by ASL. It is argininosuccinate lyase. Arginine is cleaved to release urea and return ornithine. This is catalyzed by arginase 1. This information is summarized in this slide. Please note that the first step is the rate-limiting step. CPS1 is activated by N-acetyl glutamate. This slide summarizes the major enzymes involved in urea cycle. Here is stoichiometry of urea cycle. 1 carbon dioxide, 1 ammonia, 1 aspartate, and 3 ATP are converted into 1 urea and other byproducts. 3 ATP provide a large amount of free energy to make this reaction irreversible. The rate-limiting enzyme CPS1 is activated by an allosteric effector, N-acetyl glutamate. This compound is derived from glutamate by N-acetyl glutamate synthase. This enzyme is activated by arginine. After a protein-rich meal, level of glutamate and arginine rise in the blood. Glutamate provides the substrate, and arginine is an allosteric activator of N-acetyl glutamate synthase. As a result, urea cycle is more robust. Most of urea is transported to the kidney and excreted in urine. A small portion of urea is converted to ammonia and carbon dioxide in the intestine by bacteria. Ammonia is absorbed and transported back to the liver. In kidney failure, this becomes a significant source of ammonia. Here is clinical correlation. Patients with hyperammonemia have tremors, slurring of speech, blurred vision, and coma. In severe cases, hyperammonemia may lead to death. Direct effects of hyperammonemia are twofold. First, increased level of ammonia shifts the glutamate dehydrogenase reaction towards glutamate and decrease alpha-ketoglutarate. This decreases cellular oxidation and ATP synthesis, particularly in the brain. Second, increased level of ammonia shift glutamate to glutamine. Increased level of glutamine in the brain raise osmotic pressure in astrocytes, causing brain swelling. There are two types of hyperammonemia. One is acquired, the other one is hereditary. Acquired hyperammonemia is caused by either liver disease or Reye syndrome. It occurs primarily in children with chickenpox or influenza who are given salicylate. These drugs cause mitochondrial damage and disruption of urea cycle. Treatment of acquired hyperammonemia include three methods. One is use low protein diet. Second one is use neomycin to reduce the number of bacteria in the GI tract. Third is to use lactulose. Using this sugar can lower the pH of intestinal lumen. which decrease ammonia by converting it to ammonium and trapping it in the stool. Hereditary hyperammonemia is caused by deficiency in one of the 6 urea cycle enzymes. First two conditions related to hereditary hyperammonemia are caused by CPS1 deficiency and N-acetylglutamate synthase deficiency, respectively. They both have a common characteristic, low citrulline level. Other symptoms are very typical of those with hyperammonemia, but now occurs in infants. For CPS1 deficiency, arginine is provided to activate N-acetylglutamate synthase. For N-acetylglutamate synthase deficiency, carbamoyl glutamate is provided to activate CPS1. Third condition is OTC deficiency. Similar to first two conditions, this condition also has a low level of citrulline. In addition, it has increased serum orotic acid. Due to the fact that mitochondria carbamoyl phosphate enters cytosol and is converted to orotic acid through a pathway used to synthesize pyrimidine nucleotides. OTC deficiency is the most common urea cycle disorder, and it is the only X-linked urea cycle disorder. Treatment includes low-protein diet, ammonia detoxification using sodium phenylacetate or sodium benzoate. Fourth condition is citrullinemia. Opposite to the previous three conditions, it has high levels of citrulline. In addition, it has episodic hyperammonemia. Other symptoms are typical of those with hyperammonemia. There are two types of citrullinemia. Type 1 is caused by argininosuccinate synthetase deficiency. Type 2 is caused by citrine deficiency. Citrine is an aspartate glutamate transporter on the mitochondrial membrane. Citrine transports aspartate from mitochondria to the cytoplasm. Mutation in citrine results in limitation of the substrate aspartate for the enzyme argininosuccinate synthetase. Treatment includes arginine administration to enhance citrulline excretion and sodium benzoate for ammonia detoxification. The fifth condition is argininosuccinate aciduria. It is caused by argininosuccinate lyase deficiency. It has an elevated level of argininosuccinate in plasma and cerebrospinal fluid. Similar to citrullinemia, it has episodic hyperammonemia. Treatment includes arginine and sodium benzoate. Finally, arginase deficiency is a rare urea cycle disorder. It has high levels of arginine and ammonium in the blood and cerebrospinal fluid. It also has a high level of arginine, lysine, and ornithine in urine. In addition, it has progressive spastic quadriplegia and mental retardation. For treatment, patients should be restricted to low-protein diet with a supplement of essential amino acids without arginine. Use sodium benzoate for ammonia detoxification.